What if the key to understanding antibiotic resistance isn't hidden in the future, but in the distant past? More than 3 billion years ago, long before humans existed, an ancient molecular machine was already evolving. Today, scientists can reconstruct versions of some of these ancient proteins in the laboratory. And one of them has a remarkable connection to one of medicine's biggest problems: antibiotic resistance. These proteins are called beta-lactamases. They can break apart some of the antibiotics we use to fight bacterial infections. But here's the strange part. The ancient versions weren't created in response to modern medicine. They existed billions of years before the antibiotic era. So why did scientists bring them back? And what can a 3-billion-year-old enzyme teach us about the future of superbugs? Antibiotics changed medicine forever. They transformed infections that could once be deadly into conditions that doctors could often treat. But bacteria evolve. When antibiotics kill susceptible bacteria, resistant bacteria can survive and multiply. Over time, resistance can spread through bacterial populations, and sometimes resistance genes can move between bacteria. The result is a growing problem known as antimicrobial resistance. And this isn't a distant science fiction scenario. In 2023, the World Health Organization estimated that roughly 1 in 6 laboratory-confirmed bacterial infections associated with common infections were resistant to antibiotic treatment. Between 2018 and 2023, resistance increased in more than 40% of the pathogen antibiotic combinations monitored by WHO. The problem is becoming harder to ignore. The numbers become even more striking when we look at the global burden. A major analysis published in The Lancet estimated that bacterial antimicrobial resistance was associated with approximately 4.71 million deaths in 2021. About 1.14 million of those deaths were directly attributable to resistance. And the threat is not evenly distributed. Some of the world's most dangerous resistant bacteria are gram-negative organisms capable of resisting several important classes of antibiotics. WHO's 2024 priority list places carbapenem-resistant Acinetobacter baumannii and carbapenem-resistant Enterobacterales among the world's critical antibiotic resistance threats. The question is no longer simply, can we invent another antibiotic? It's also, can we understand how resistance evolved in the first place? Here's where the story gets surprising. Antibiotic resistance did not begin with the invention of penicillin. Nature was already producing antibiotic-like molecules long before humans began making drugs. And microorganisms evolved mechanisms to survive them. Beta-lactamases are one of the clearest examples. These enzymes can break the beta-lactam ring found in an important family of antibiotics. Penicillins and many cephalosporins belong to this family. So when a bacterium produces a beta-lactamase capable of destroying the drug, the antibiotic can lose its ability to do its job. Scientists now believe some beta-lactamase lineages are more than 2 billion years old, which means the evolutionary story of antibiotic resistance is far older than modern medicine. But there was a problem. Scientists cannot simply travel back 3 billion years and collect an ancient enzyme from a laboratory freezer. So they built a molecular time machine. It's called ancestral sequence reconstruction. Scientists compare the DNA, or protein sequences of organisms alive today. They use evolutionary relationships and statistical models to estimate what ancestral sequences may have looked like. In other words, they work backward through the evolutionary tree. They don't recover the original protein from ancient rocks. They reconstruct a plausible sequence. Then they synthesize the gene, produce the protein in the laboratory, and test what that reconstructed molecule can actually do. And that's exactly what researchers did with ancient beta-lactamases. In a 2013 study, researchers reconstructed several ancient nodes in the evolution of Class A beta-lactamases. The evolutionary nodes they targeted reached back roughly 2 to 3 billion years. The scientists then synthesized the inferred proteins and studied them in the laboratory. The results were unexpected. These ancient proteins folded into the characteristic beta-lactamase structure, and they were remarkably stable. Their melting temperatures were roughly 35 degrees Celsius, higher than those of many modern beta-lactamases used for comparison. But stability wasn't the most interesting part. The ancient enzymes could also degrade several different antibiotics in laboratory experiments. They weren't specialists. They behaved more like molecular generalists. Think of it like this. A modern specialist might be extremely good at one specific job, but an ancient enzyme may have been more flexible. It could interact with several related molecules, even if it wasn't perfectly optimized for any single one. This property is called substrate promiscuity, and it may help explain something fundamental about evolution. A molecule doesn't always evolve by becoming completely new. Sometimes evolution starts with a flexible generalist and gradually turns it into a specialized tool. In later work, researchers found that differences in the internal movements of these proteins could help explain how ancestral generalists became more specialized modern enzymes. The shape of a protein isn't the entire story. How that protein moves may matter too. And now we reach the paradox at the center of the story. These ancient enzymes were not created to defeat antibiotics invented by humans. Humans didn't exist. Modern pharmaceutical antibiotics didn't exist. Yet the reconstructed proteins could interact with several antibiotics that we use today. That's not because bacteria somehow predicted modern medicine. It's because some antibiotics resemble chemical structures that existed in nature long before humans turned them into drugs. Evolution had already been experimenting with these molecular battles for an unimaginably long time. We didn't create the arms race. We entered one that was already ancient. So could these ancient enzymes become a new weapon against superbugs? Not yet. And this distinction matters. The 2013 experiments did not produce a human treatment. They were laboratory studies designed to understand the evolution, stability, and function of ancestral beta-lactamases. In fact, these enzymes themselves are resistance mechanisms. Their ability to break down antibiotics is exactly what makes beta-lactamases dangerous when bacteria use them against modern medicine. So the real opportunity may be different. Instead of using the ancient enzyme as a drug, scientists can use its structure and behavior as a clue. Understanding how these proteins work may help researchers understand how resistance evolves, and potentially design strategies that avoid, block, or outmaneuver those mechanisms. And the race is continuing. The World Health Organization's current priority list identifies resistant bacteria for which new treatments are urgently needed. At the same time, researchers are investigating new antibiotics, new targets, biological approaches, diagnostics, and other ways to slow or overcome resistance. WHO's 2025 review of the antibacterial development pipeline evaluates candidates in clinical and preclinical development against these priority pathogens. But there is no single magic solution. New drugs can eventually face resistance too. That's why understanding the evolutionary machinery behind resistance matters. If we can see how bacteria adapt, we may have a better chance of staying one step ahead. The most fascinating part of this story isn't really the age of the enzyme. It's the idea behind it. Evolution has been running experiments for billions of years. Every organism alive today is the result of an enormous history of molecular trial and error. By reconstructing pieces of that history, scientists can turn evolution into something we can study experimentally. We can ask, what did an ancient protein look like? What could it do? How stable was it? And how did a flexible molecular machine eventually become the specialized systems we see today? The past isn't just something we study in fossils. Sometimes, The past is still encoded in the molecules of life. A 3-billion-year-old protein cannot tell us exactly what the future of medicine will look like, but it can teach us something important. The biological world has been solving and creating molecular problems for billions of years. And sometimes, understanding the future begins by looking backward, not to find an ancient cure, but to understand an ancient battle. The past may not give us the weapon. It may give us the map. This is Quantum Mind, stories that explain our world.